Good morning, students. Today I am going to discuss about enzymes in genetic engineering, a genetic engineering course for MSc biochemistry students. Before going to start enzymes, an introduction. The ability to manipulate DNA in vitro depends entirely on the availability of purified enzymes. All enzymes are protein, but all proteins are not enzymes. The DNA molecule in specific ways. At present, no chemical method can achieve to ability to manipulate the DNA in vitro in predictable way. The only enzymes are able to carry out the functions of manipulating the DNA. Each enzyme has vital role to play in process of genetic engineering. The enzymes which is important for the commercial application. Various enzymes used in genetic engineering. What are the various enzymes? Nucleases, restriction enzymes, DNA ligases, kinases, phosphatases, especially in alkaline phosphatase, reverse transcriptase, terminase, deoxynucleotide transferase, RNAsP. One of the example of enzymes, such manipulation of DNA are conducted by toolkit of enzymes. For example, restriction endonuclease are used to molecular scissors. This figure shows the cutting of DNA you are using for the restriction enzyme. DNA ligases function to bond species of DNA together. That is, a ligase means joining. A variety of additional enzymes that modify the DNA are used to facilitate the process. What are the main enzymes used in DNA? The enzymes are phosphatase, the attack polymerase for PCR reaction by thermocycler, nuclease, reverse transcriptase, DNA ligating enzymes. These are the enzymes are used in DNA. Nuclease. First one is the nuclease. The nuclease function of various enzymes is used in genetic engineering are described a group of enzymes which cleave or cut genetic material, either may be DNA or RNA. DNAs and RNAs nucleases are further classified into two types based on the substrate on which they act. Nucleases which react on cut DNA are classified into DNAs, where those which are non-RNA are called RNAs. Whatever we need for DNAs or RNAs. Another one important enzyme is called the restriction enzyme. The restriction enzyme is important for cutting up the particular DNA. This will be DNAs which react on specific position or DNA sequences on the DNA are called the restriction endonuclease. The RE indicate the restriction enzyme as a recognition of sequences or restriction site. These sequences are palindromic sequences, different restriction enzyme present in the different bacteria that recognize the different or same restriction sites. This will be cut into two different points with the restriction site. Such restriction enzymes are called as the isosomers. Interestingly, no two restriction enzymes from the single bacterium will cut at the same restriction site. What are the examples? The different types of restriction enzyme: type 1, type 2, type 3. Different enzymes used for the cutting of DNA to generate the blend ends with a short single strand over the hand of another each end. The restriction enzyme have three important features. It make breaks in the polymeric sequences. The break are usually not directly opposite to the another. The enzymes generate to DNA fragments with the complementary ends. Another one important enzyme, DNA ligase. Ligase means it is the joining of the particular DNA together. The recombinant DNA experiment requires to the joining of two different DNA segments or fragments in vitro, but the segments unrelated that are weak do not withstand the experimental condition. The DNA should join by the enzyme called ligase. The DNA ligase joined the DNA molecule covalently, catalyzing the formation of phosphodiesteric bond between the adjacent nucleotide. DNA ligases isolated from the bacteria E. coli or T4 bacteria phage is widely used. These ligases more or less catalyze the reaction in the same way at a different only in the requirement of the cofactor of enzymes. Kinases. Another one is an important enzyme group of enzymes is called as a kinase. The adds a pre-pyrophosphate to a wide variety of substrate like proteins, DNA, and RNA. It uses ATP as a cofactor add to the phosphate breakdown to ATP and ATP hydrolysis. It is used for molecular biology and genetic engineering to add the radiolabeled phosphatase. Another one important enzyme is called as alkaline phosphatase. Phosphatase are this alkaline means there is a alkaline condition above 8 pH. The pH which react to the buffer, basic buffer called alkaline phosphatase. Most commonly bacterial alkaline phosphatase BAF, calf intestinal alkaline phosphatase and shrimp alkaline phosphatase are used in molecular cloning experiments. This alkaline phosphatase is a metalloenzyme and has zinc ions under them for the activating site. Another one important enzyme is called as a reverse transcriptase. This enzyme uses an RNA molecule to say template and synthesize a DNA strand complementary to a RNA molecule. These enzymes are used to synthesize the DNA from RNA. These enzymes are present in the most RNA tumor viruses and retroviruses. This will be helpful for DNA dependent DNA polymerase. Reverse transcriptase enzyme after the synthesis the complementary strand the nucleotide with the help of DNA and extra nucleotide stretch without complementary sequence. This sort is called as a R loop. Another one important enzyme is called as the terminal deoxynucleotide transferase. These enzymes is a polymerase which add to the nucleotide 3OH end, but it does not require to any complementary sequence and does not copy any DNA sequence unlike clone fragment. The terminal in deoxynucleotide transferase added to nucleotide, it comes to active site, it does not show any preference for the any nucleotide. Another one important is some RNase. RNase P is specifically cleaved at 5 end of RNA. It is complex enzyme consisting of small proteins and at the 377 nucleotide of RNA molecule. It has been observed RNA molecule possesses at least part of enzymatic activity in complex. Hence, it is enzyme is a ribozyme. DNA polymerase. This is important for polymerase reaction. The polymerase is an enzyme that makes a new DNA atom to strand, but both prokaryotic and eukaryotic contains more than one species of DNA polymerase enzymes. Only some of the enzymes actually carry out the replication. Sometimes they are designed to DNA replicates, and the other are involved substituted roles replication and participate to repair synthesis of DNA to replace the damaged sequences. The figure shows for the template and primer requirement of DNA polymerase are enzymes that catalyze the synthesis of new DNA strand from the pre-existing strand. This is a template strand from the 3 to 5 regions. What are the applications of restriction enzyme? Traditional cloning is a robust technology to day by day experimentation in molecular biology laboratory. DNA mapping studies include identifying genetic disorder, loci, assessing the genetic diversity of population and parental caring, understanding the epigenetic modification. Nowadays, very famous research is going on epigenetics with the modification of DNA that are heritable changes. What are the heritable diseases associated with the epigenetic studies? Application related to genetic engineering, in particular DNA. In various applications related to the genetic engineering, DNA is cleaved by using the restriction enzyme. They are used to insertion of DNA genes to plasmid vector during the gene cloning and the protein expression. Restriction enzymes can also be distinguished gene allele by specific recognizing single base changes in DNA. This only possible to the mutation alteration the restriction site present in the allele. Restriction enzymes are used to restriction fragment length. That is one of the technique RFF, restriction fragment length polymerism analysis for identifying individuals or strain in the particular species. Thank you.